Okay, thank you very much. I'd like to thank the organizers for inviting me and all of you for getting up early for this morning session. So the first slide kind of summarizes most of the talk and has some of the take-home lessons. The first one is, this is just taken from BioWorld Today that had a little article after we published some of this recently in the fall. And the key point here is that I'm going to tell you about the work we've been doing to achieve gene editing by IV administration. So what we're working to do is develop a strategy for in vivo gene editing, and I'll show you some of the data in a mouse model of thalassemia where we've been able to achieve some evidence of that. And the second thing is shown in the middle there. This is a diagram of the DNA triple helix. That is the basis of the work we've been doing. And the key point I want to make here is that the formation of a triple helix, which is sequence dependent with a synthetic DNA analog, triggers DNA repair. So in some ways, once it does that, it's conceptually similar to the CRISPR-Cas9 and other nucleases that directly generate strand breaks, but then their effects are only mediated once DNA repair occurs. So I have some disclosures. I'm a consultant and have some equity in 2 companies as listed there. So I mentioned the many nucleases that people are working on, and as you can't escape hearing about in this meeting, they work by making strand breaks, and I'm sure you're all familiar with these. So a DNA triple helix forms in a sequence-specific way, but it's in some ways much more complicated than, for example, the guide RNA for CRISPR. And the reason is first of all, triplexes only form at regions of purines on one strand and pyrimidines on the other. And depending on how many A's versus how many G's, you can have 2 different motifs, a pyrimidine motif or a purine motif as illustrated there. And there is a binding code that is based on Hoogsteen hydrogen bonds, which are illustrated in this diagram. I'm having a little trouble getting the cursor to work, so I'm maybe not going to keep pointing to things. But as you can see there, there's a binding by Hoogsteen bonds, and this occurs in the major groove of the duplex. The important point is this is sequence-specific. And interestingly, I gave this seminar a number of years ago at the University of Utah, and it turns out triple helices were first discovered in 1957 by Alexander Rich, and the paper was Felsenfeld, Davies, and Rich. But there was an undergraduate student at MIT who worked in the summer and did a lot of the grunt work for that paper, and he came up to me at the end of my seminar at Utah and just say, you know, I did some of the early work on triplexes, but I didn't get my name on the paper, and that was Mario Capecchi. So anyway, it's a small world. So the idea, as I've shown you, is that we have evidence that formation of a triplex disrupts the helix of the DNA and triggers repair. And so the idea would be you trigger repair in the vicinity of the mutation that you want to edit, and if you provide a template DNA, or we call it a donor DNA, we use a short oligonucleotide around 60 nucleotides that can mediate recombination or information transfer to modify the sequence. So this is work from almost 20 years ago, and this is when we first demonstrated that the formation of a triple helix would trigger DNA repair. So what you can see here is just a plasmid substrate in whole cell extract from HeLa cells that reconstitutes DNA repair pathways. And the point is that the incorporation of radioactivity in the plasmid is indicative of DNA repair. So anyway, what's the big deal with triggering repair or making DNA strand breaks? So many of you know that early work in the 80s and 90s showed that when you introduce foreign DNA into mammalian cells, it integrates only about 1 time out of a million in the cognate place. Most of the time it goes in an ectopic location. And that's why, speaking of Mario Capecchi, Capecchi, Evans, and Smithies won the Nobel Prize for developing a selection technique to find the place where it integrated correctly. But other people in the field, such as Maria Jason, John Wilson, and Dana Carroll, showed that if you could trigger DNA repair or make a strand break at the target site, you could greatly increase the efficiency of recombination in the cognate site, and that really triggered the race to develop targeted nucleases. But then around that time, we showed, like I said, that the formation of a site-specific triplex would trigger DNA repair and potentially could do the same thing. So we began a race to find DNA analogs that would improve the efficiency of that system, because our early work showed frequency of gene modification that was in the range of 1 to 10,000 to 1 to 1,000, which was not clinically meaningful. So after surveying a lot of analogs, and actually I did some work early on with Mano, who's going to be giving a talk later, but also in collaboration with Michael Egholm and Peter Nielsen, and more recently with Dan Atlee at Carnegie Mellon, we focused on peptide nucleic acids, which feature nucleobases positioned to bind to DNA by Watson-Crick, and it turns out they can also form triplexes. But as you can see there, the PNA has an uncharged polyamide backbone, which turns out to be advantageous for high-affinity binding. So PNAs not only can make a triple helix, but they bind so well to DNA they can do strand invasion. And what you see here is a design of a PNA-DNA-PNA triplex on the left with displacement of the other strand of the DNA, sometimes called a P-loop. Then, because the Hoogstein bonding portion of the PNA is limited to the polypurine run, and typically in most genes you can't find more than 10 to 12 in a row, to achieve more specificity, we developed an approach to extend the Watson-Crick portion with a tail. We call this a tail clamp design, and this helped improve the specificity and the binding affinity, and was also based on work out of the labs of Peter Nielsen and David Corey. So you can see at the bottom there how the sequence might line up, and you have a little linker region to connect the two domains, the Watson-Crick and the Hoogstein, to form the tail clamp. So the idea here is like a DNA triple helix, if you form this PNA tail clamp on DNA, it triggers DNA repair, and we actually published that this structure triggers repair even more strongly than a DNA triple helix, and you can mediate you can catalyze gene editing. So then in recent years, our work has been advanced by two key collaborations. One is on the left, the use of nanoparticles consisting of a biocompatible matrix called polylactoglycolic acid, PLGA, which is actually in use in people now. It's FDA approved as a drug delivery material. And then the use of a modification of a PNA called a gamma PNA, which you can see there on the right. It's actually more officially called mini-PEG gamma, because you see two ethylene glycols coming off of the gamma position on the polyamide backbone. And if you notice, there's a little R in parentheses there, and that's a hint that that forms a chiral center. So what does that mean? If you make these in a stereoregular way, these gamma PNAs have superior properties compared to regular PNAs. So regular PNA, as shown on the left there, you have a disordered hydrophobic molecule. But when you make a gamma PNA, the substitution forces the molecule into a pre-organized right-handed helical structure, which is just right for binding to RNA or DNA. And you can see in the lower left that circular dichroism analysis shows that this molecule is helical by itself in solution. And then the melting curves on the right show that gamma PNA DNA hybrids are stable up to almost 90 degrees and gain about 30 degrees in stability compared to PNA DNA hybrids. So that demonstrates the improvement that is achieved with this substitution. And then here is a diagram of how these nanoparticles are made. This is done in the lab of Mark Saltzman at Yale in biomedical engineering. And the important point is that you can encapsulate the PNA and the donor DNA together in a single nanoparticle. And this is highly biocompatible and highly efficient for delivery via intravenous administration to multiple tissues. And Mark's lab has demonstrated this not only with these types of molecules, but with siRNAs and other types of nucleic acids. So now I want to tell you about how we've applied this to beta thalassemia, and most of you are familiar with this disease of hemoglobin production. So I was very lucky a number of years ago to have an excellent excellent postdoc come to my lab, Linda Gorman, who had been a graduate student with Richard Cole at the University of North Carolina. And she suggested that maybe we should use this really interesting model that Richard had developed in which the green fluorescent protein was engineered to contain the intron 2 from the beta-globin gene, but containing a thalassemia-associated mutation that created a cryptic splice site. And this turned out to be very useful for us to develop this system. Now I'm going to tell you a little bit of work we did with this green mouse, as Richard called it, but then we're going to move into a true mouse model of disease model of thalassemia. So the idea here is, you can see on the left there, taken from Richard's work, if you have an oligonucleotide that alters the splice site choice, you can lead to expression of a correct GFP, but you can also do that by editing that site. So it's a very versatile system to test different approaches with oligonucleotide therapy. We went ahead and designed some tail clamp PNAs to bind to that the intron in the vicinity of the mutation to be corrected, and you see some examples there of the sequences and some data showing their binding specificity. So we went ahead and formulated those molecules with the donor DNA into nanoparticles. Then we took bone marrow cells from the GFP beta-globin fusion mice that Richard had sent us, and we treated those bone marrow cells in culture, and then subsequently performed flow cytometry to quantify the GFP positive cells. And we found in this particular initial experiment, after one exposure to the nanoparticles, 1% of the cells had turned green with PNA1, and so that looked to be a superior sequence, and we proceeded to work with that. The next step was to advance that with the inclusion of the gamma modification, and that's shown here. In this case, we modified every other residue with gamma. We're in the process now of comparing a lot of different variations on this, but this is not trivial because these are not commercially available, and in fact we make these all ourselves. I had actually bit the bullet, and several years ago I recruited and now have 3 PhD chemists in my lab, because basically every advance that I've made since really the original discovery of this concept has been by making friends with chemists. So that's really a key. So we made these, and we tested the gamma modified versus the regular versus the scrambled. And you can see here that there's a boost in editing, looks to be about 2-fold in this experiment. This is ex vivo treatment of bone marrow, but in fact in the next few data I'll show you, it's usually about 3- to 4-fold increase with the gamma modification. So this was promising. So then I had a very good graduate student in the lab, Nicole McNear, who had the idea of, well, let's actually fractionate the cell populations and see if there's some difference in editing. And this is what we found. So this graph has two punchlines. One is that it's showing that the gamma modified PNA that's on the right is about 3 to 4-fold more active than the regular PNA. But also the CD117 positive cells were the ones being edited in the bone marrow. And as some of you know, the CD117 marker represents the c-KIT, the KIT receptor, which is present on most bone marrow stem cells. So what you see here is ex vivo treatment of the whole bone marrow. Then the cells were stained and double sorted for the marker and then for the GFP. And you see in the CD117 there was 9% correction in a single treatment. We actually also did it the reverse. We pre-sorted the cells and then treated them. You see here about 7% modification in a single treatment done ex vivo. So what's going on? Well, it turns out that c-Kit is stimulated by the ligand stem cell factor. So actually, if you add stem cell factor to the media, you get a further boost in editing, and you can see on the left there up to about 15% in a single treatment. Or if you inhibit the KIT pathway with tyrosine kinase inhibitors, you can reduce the gene editing. So there's some evidence there that the KIT pathway is important for editing in these bone marrow cells. And one of the reasons we think that may be going on is that if you sort the cells for the presence of the KIT receptor, the CD117, the positive ones have a higher level of expression of many of the DNA repair factors that would be expected to play a role in the processing of the triple helix lesion. Similarly, if you treat those cells with stem cell factor, you boost the expression of DNA repair even more. And that's shown here in the red. So now we took this to a mouse model of thalassemia, and we also obtained this from University of North Carolina, and this was originally made by the authors you see there, including Richard and Oliver Smithies. And in this model, the mouse beta major and minor loci are replaced by a human beta locus containing that same thalassemia-associated mutation. So this is the human beta-globin gene replacing the mouse beta-globin gene. And so these mice have a version of thalassemia. I should say that the homozygous mice for this mutation are not viable. So what you end up working with are their heterozygotes, but they still have a version of thalassemia with low hemoglobin counts and other problems. So we took these mice and we treated them with the nanoparticles containing the gamma, PNA, and donor DNA that I showed you earlier. In this experiment, we did 4 treatments, 2 milligrams of nanoparticles given IV and 150 microliters each, given every 48 hours for 4 treatments. And then we waited up to 20 weeks. We looked at the mice at different intervals. The earliest usually was about a month, but we actually carried this out over 140 days looking at these hematologic parameters. So this is one of the key results, and this is a measurement of blood hemoglobin. And what we found is by the 30-day time point, the mice showed elevation of the blood hemoglobin into the normal range, and this was sustained without further treatment for 140 days, after which we stopped analyzing the mice, because at some point it becomes harder and harder to obtain blood from the animals because you don't have any venous access due to sclerosis. So basically, we can say there's a sustained elevation of hemoglobin. If you look at the reticulocyte count, and this was measured, I believe, after a month or so, this is 36 days, you can see that in the treated mice, the reticulocyte count goes back down to normal by 36 days after the end of the treatment. Now, what about the splenomegaly? As many of you know, in thalassemia, there's to a large extent extramedullary hematopoiesis, which affects the spleen. You get enlargement of the spleen, as shown here in the blank treated animal, and in the other controls. But in the gamma treated mice, on average, the spleen size was substantially reduced, and you see the quantification of spleens from several animals at the bottom there, but you can see that visually. So there was substantial reduction in the splenic mass. There's also improvement in the splenic architecture. So in the top picture, you see a normal spleen with the typical white and red pulp architecture. The blank nanoparticle on the lower left is basically untreated mice, And you can see a disorganized spleen because of the extramedullary hematopoiesis. But then in the gamma PNA-treated mice, you start seeing a return to the demarcation of the red and white pulp in the spleens. And that's consistent with the reduction in the splenomegaly. Now, what frequency of gene editing did we achieve? Well, it turns out in total bone marrow, it was, as you can see here by deep sequencing, about 4% of the total bone marrow cells. So this is not separating out any particular marker. We found about 4% gene editing. When we broke it down by lineage, and this is with advice from people who are aficionados of hematopoietic progenitor cells, we saw a slightly increased frequency of editing in putative hematopoietic stem cells demarcated by the markers you see listed there, up to a frequency of about 7%. So this was after the 4 intravenous treatments of the mice. Then we asked, what about human CD34 cells? So we took human cells from these are from a repository that are left over after autologous transplants. And basically, these are not thalassemia-afflicted cells. So we actually, in this case, are putting in that same mutation using a different donor sequence, which makes the mutation rather than corrects it. And treating those cells ex vivo, you can see on the left side of the bar graph, we achieved about 5% gene editing in a single treatment. We did include SCF treatment of these cells in addition to the typical stem cell medium. We then transplanted the treated cells into NOD-SCID mice and then let them engraft. And then 8 weeks later, we harvested bone marrow from the cells, from the mice, and sorted the human CD34 cells. And among those cells, we still detected evidence of gene editing at a frequency of about 3%. So what about off-target effects? So one of the things we did is we've looked at the production of strand breaks, which can be indicative of genotoxicity. And we've done that not by trying to capture them with an oligonucleotide, but by a method that people who study DNA repair like to use, which is looking for foci in the chromatin of a marker called H2AX. So what happens is that histone component gets phosphorylated on a serine by the damage response proteins ATM and DNA-PK when there is a double-strand break. So you see foci forming in cells that have experienced double-strand breaks. So in the lower right panel, that's indicates 5 gray of ionizing radiation, you see a classic picture of gamma H2AX foci indicating many double-strand breaks. In contrast, in the upper right panel, you see in cells treated with the gamma PNA and donor DNA nanoparticles, you don't detect any increase of H2AX foci above background, and the background is in the panel on the upper left. Interestingly, and I'm just going to show this data because I don't want to, I don't have too much to say about this right now, except that the lower left shows what happens to those same cells when you introduce the Cas9 enzyme without a guide RNA. When you add the guide RNA at the same time, the next panel over shows a substantial reduction in induction of double-strand breaks, so that's something that might be worth pursuing. And that's quantified here, but in the interest of time, I'm going to move on to just point out we also did deep sequencing of the most closely homologous sites in the genome to see if there were other mutations made at those sites. And as you can see here, in a situation where the beta-globin target underwent 4% gene editing, this is in the mice that were treated intravenously. The frequency overall in the off-target sites out of about 8 million reads was about 1,000-fold lower, although there was one site that seemed to crop up as maybe conducive to some effect, as you see there. In the human cells, interestingly, we saw almost no sequence changes in the closely homologous sites, so that the off-target frequency out of about 25 million reads was 100,000-fold lower than in the target site, which was at about 5%. In the treated mice, we didn't see any early or late evidence of inflammatory immune response, although we have not looked for the production of antibodies that might come up, but we know that the PLGA material itself is not immunogenic. So that's where we stand now. We're in the middle of studies trying to apply this to a mouse model of sickle cell disease. We have been working on cystic fibrosis, and we had some success by intranasal administration of the nanoparticles into a mouse model of cystic fibrosis, which with some improvement of the potential difference of the nasal epithelium, as well as some sequencing evidence of gene editing. And also we're working with Pat Gallagher at Yale on hereditary spherocytosis, for which we also have a mouse model. So I'd just like to acknowledge the people that did the work. Most of the beta-thal work was done by Rahman Bahal, who's an excellent postdoctoral fellow in my lab. He actually is one of the chemists who got his PhD at Carnegie Mellon, and he's on the job market, so if anyone is looking for a very excellent young junior faculty, let me know and I'll put you in touch with him. And I'd just like to acknowledge collaborators. Mark Salzman, I mentioned, Marie Egan on the cystic fibrosis, and Pat Gallagher and Diane Krause are hematology experts that have helped with the mouse model, and then Daneth Lee, who developed the gamma PNAs. And I'll stop there. Thanks.
So if you have any questions, please just come to the microphone.
So are there any differences between gamma PNA and regular PNA, how it's handled inside cells, how long time it survives, and things like that?
We don't know the answer to that. We haven't measured any pharmacokinetics. Both regular PNA and we think similarly gamma PNA are resistant to nuclease degradation because they're not normal substrates, but we don't know how quickly they're cleared. As far as inducing repair, we haven't directly compared the level of repair activity in vitro. We do know the binding is better.
So you mentioned that the homozygous mice do not survive. So would it be reasonable to try to do like in utero treatment of the mice and see if you can maintain?
So I have an excellent grad student, Adele Ricciardi, who actually was an undergrad at UNC. So that's a lot of good people come out of UNC, and she has been doing that. And we have some positive results, and we have a paper in review. She got together with a pediatric surgeon, David Steitelman, who was expert at, the microsurgery necessary to do in utero treatment in mice. So that story hopefully will come out soon.
And the second question is, so besides PNA or modified PNA, have you tried other oligos, like say LNA?
Yeah, so I glossed over one slide. So we kind of wandered in the wilderness for about 10 years before we got to the gamma PNAs, or got to PNAs and then gamma PNAs, and we have tried more than 50 different analogs. I mentioned I work with Mano, I've worked with Michael Seidman, worked with Kia Watanabe, quite a long list of excellent chemists, as well as using commercially available molecules like LNAs, which we found that LNAs were not particularly good at this, forming triplex. And also one of the advantages of PNAs, they don't have a charged backbone, so you can make that PNA, DNA, PNA triple helix, whereas with the charged backbone molecules, you really only get the triple helix.
Peter, great talk. I have two questions. One is, do you need the gamma PNA uniformly modified, or you can sprinkle it? Because it's basically the hydration and solubility. That's one question. The second one, in this entire technology, do you need a nuclear targeting signal for this?
Yes.
That's one of the triple helix issues from the early times.
Yeah, so we think you probably don't need uniform modification, that some number is enough to give you that pre-organization as well as the improved solubility. And so we're just now, we have enough material now to test a lot of variations, so we're going back to do that. We don't have a nuclear targeting signal. We're just using the nanoparticles for delivery. You know, Mark Saltzman's work with labeled nanoparticles shows very good in vivo uptake in bone marrow and liver in multiple tissues. And you can see contents in the nucleus. So we think that, and in fact, we followed labeled PNAs, so you can see them in the nucleus. Now, it may be they would be even better with a nuclear localization signal. One of the challenges is these are long PNAs, and you know, you will know as a chemist that if you start making these fancy PNAs and then add a peptide sequence, it becomes a challenge for synthesis unless you did sort of a post-synthetic click chemistry or something like that.
Just a quick question. So you mentioned LNA. I was just wondering, LNA forms triplexes, but is it that LNA didn't form triplexes, or was it that the repair system was not induced when you used LNA?
So empirically what we found is the effect of the LNA substitution was not better than standard DNA or phosphorothioate protected DNA. In general, this work I did with Mike Seidman a number of years ago, in general we found that the RNA-like molecules did not trigger repair as well as the DNA-like molecules.
Actually, I have a quick question. So do you think with the percentage of correction that you get, would that be sufficient to treat a patient? So what would be the minimum amount of hemoglobin that you need for reverting a phenotype?
Yeah, I think most people say, you know, like hematology settings, that at least 15% is needed for these types of diseases, 15 to 20 for something like sickle cell. I think we were a little bit lucky in this mouse model because the corrected red blood cells have such an advantage in terms of lifetime. The turnover of the thalassemic cells lifetime is so short that you may only have, you know, 5 to 7% in the bone marrow, but in the periphery, you know, because the lifetime is extended and the sick ones are dying out, you get more bang for your buck.
Yeah, thank you. All right. Thanks.
Thanks.